Hello, I'm Professor Brown, and we're going to go over how to do what's commonly called the Kirby-Bauer test to test the effectiveness of antibiotics or antimicrobial drugs on particular bacterial species. So much like with the chemical disinfectant test, we have a nice large agar plate that I've already labeled. In this case, all I really need is the date, my initials, and the bacterial species. We don't need to label the plate with the antibiotics because we're going to be using pre-made antibiotic discs that are actually already labeled, so the label will let you know what it is. So I've already swabbed the entire surface of this agar plate exactly as we did with the chemical disinfectant disc diffusion test. So the entire surface has been swabbed in this direction, and in this direction. So the entire surface has been inoculated with E. coli. Now all we have to do is transfer discs with various antibiotics to the surface and incubate the plate and see what kind of results we get. And as with the chemical disinfectant disc diffusion test, it's very important to make sure that the discs are placed in such a way that they're as far apart from each other as possible. So I'll demonstrate that as we go along. So we're going to use our forceps, and we're going to have to dip them in alcohol and briefly flame them before we get our first antibiotic disc. So what I have here is streptomycin, and I'm going to take this little device here. We'll push the disc out, and then I can grab the discs with the forceps. And this one I'm going to place right smack in the middle. And then just gently press on it to make sure it's nice and flush against the agar surface. And then sterilize my forceps and get the next antibiotic. Now this one doesn't have one of those little metal ejectors. Let me get a different one here. All right, so we have erythromycin here. And once again, use this little device to push the disc, grab it with the forceps, and I'm going to place this one about right here. Gently press and so forth. So I'm going to do that for another 6 antibiotic discs, and when I'm finished, you'll see how they're arranged in such a way that they're as far apart from each other as possible. All right, now as you can see, I've placed a total of 7 antimicrobial drug discs on the surface of this agar plate, and hopefully you can see that I arranged them so that there's as much space around each disc as possible. That's important because what you're going to look for next time you come to lab is you're going to be looking for a zone of inhibition, a circular zone of no bacterial growth around each disc, which you're going to measure, and you want to make sure there's as little overlap as possible between each disc. So, now that we've got this done, I'm going to take the plate and, as usual, turn it over, lid down, which is how we want to incubate it. Again, that's for the same reason as before. We don't want to risk having any condensation dripping on the agar surface. That'll basically ruin the experiment. And as you can see, the discs stay put, so they're not going to fall down. They're going to stay right where they are, and everything will be just fine. Okay, and then all you got to do now is incubate your plate for 18 to 24 hours at 37 degrees Celsius.